Supposed to add this to all of them.
Water.
Wait, no water.
Yeah, so the final volume will be 8 milliliters. I guess 2 through will be a little bit higher because there's a little bit of acid in it. But you could think that if they had 5 drops of water, if it's 1 and 2 on a whole volume constant.
Yeah, your favorite video. You want that like that? Like, are you taking pasta? Yeah. You do well in the upper view. Yeah. That is so bad. I don't even know why. It's not like the way I like. Yeah.
Did you get it?
I'm at 24.
Yeah, that's what I said I did.
Dawn got a 30 somehow. Yeah.
Okay, well, Dawn's like really open.
Dawn loves the movie. It's not a history, the averages are not history, like, I just find.
I'm sure trying to be like, no, like, Dawn is just like good at that kind of stuff.
No way, he's definitely like paying the teacher. Yeah, not tax.
I did so.
Yeah, I'm definitely reading that. Send us an email, like, you can retake them, I'm like, oh.
For full points.
I have no idea. Probably.
Like, I'm fine with like half points for like corrections.
Yeah, so.
You should boil it all at once.
How do I know it?
You'll be here forever.
Put it in order.
Or you can hold them. It's only a few minutes, so if you put them all in together, just put them in together in order.
How does this not fall?
What's the worst that can happen if it
Be mildly annoying.
one break, 8 sessions. So.
Oh, wait, I thought you were saying 8 years, and I was like, no way. I'd be a miracle.
Actually, one thing to
I
say,
had notes when I started off. So maybe that's how it was. Well, I don't know when I started.
senior college admission, at least 3 minutes. Let me tell you a little something about that. Larry never learned how to tell time. You can have a bell schedule on your campus. If so, you're going to be in trouble.
Zach, we're literally creating a next like styrofoam on the hot plate and we have to leave.
Oh my gosh.
In 8th grade, I wrote a tech deck across the hot board, and Mr. Crook got so mad at me.
I think I remember.
And it melted the wheels off.
The important things. It was awesome, even though it was extremely toxic.
I could be the only person in the world that I could tech deck across.
Pretty sure John Hagner did that.
Yeah, he did. It was me and Sean. We were in the back, just like, There's this new study mode.
Usually it's kind of old, but
Definitely an inferior. Yeah, a minute and a half.
I'm gonna bring everything to the weight. Open the orange, why did you do?
I think I probably went over.
45 seconds.
Mr. Bergeron, what does a hot bath actually do?
Well, I don't want to spoil it too much, but I mean, all chemical reactions require an activation energy, right? That needs to be overcome. Well, no, even non-spontaneous reactions have an activation energy barrier, right? So sometimes there's enough energy within the system, but sometimes there isn't. So we're giving it some.
Okay. So it can turn heat energy into, I don't know, whatever energy it needs to react.
Yes. Yes. The molecules are going to.
Oh, because they just speed up. Yeah, I remember. I said I needed to count. It's not the activation energy. Wait, I think the activation energy line just like moves, right? Just temperature.
Things go down. Magnitude increases would be a better way to say.
I'm thinking of a different graph. It's like the bell, and it shows like how many react under certain temperatures, and that's like the EA. But yeah.
What's it called? Solution to living?
Yes. You remember the concept?
Describe with your parents.
Yeah.
Oh,
How it is coming out.
Okay, well, I know it's different, so.
No. Mr. Bergeron, can I store this back in here? Or not?
Yeah.
Yeah, it's in the middle one.
Yeah.
Then why are you doing so? That's like different font.
I have no fun. Yeah, I don't know.
Oh, whoa!
Yeah.
I know, I dressed up, but.
Mine and my other one is the same thing. That's so sad.
Here we go. I thought I had a different Allen Walker or something, but obviously I don't know.
Oh, it's pretty yellow.
Wait, isn't it a fine color?
No, for the,
No, I didn't know I was wrong. I don't think this.
Like, just like, just pour the whole HCl in there. But
Drink the HCl.
That's it.
do we dissect stuff in this class, Mr. Bergeron?
No, not really.
I feel like it'd be kind of fun.
Organ systems went away about a decade ago.
Really?
Plus, there's a pretty big push against using animals for dissection of organs. Very, very rarely do rookies have the time and the patience to dissect carefully. You end up creating what's a nice way of saying this?
State of disorder.
There you go. Yes, it's a corpse in the full throes of entropy. But we don't have an animal.
Sharp. Sharp, really.
What level is this?
Yeah.
Right, so as soon as you are finished, by finished, I mean you have something to see. You take a picture. You can turn off your hot plates because you're the last class.
Wait a lot.
I'm trusting and confident that you will figure out how to sum it up.
Dear college visits, let me tell you something about the entire 11th grade class. The odds tend to confound them.
Got it, I swear.
Yeah, so once you have your picture, you can clean everything up. The tubes can be washed out with tap water. Pipettes can be rinsed off with tap water. There's plenty of sinks. There's 3 up here, there's 1 in the back, and 1 in the front room that you can use if you'd like.
Yeah, I don't mind it. Look at it. Zach, mine looks more edible than my pink one looks like a good.
All right, folks at home, I hope you can hear me okay. In 3 or 4 minutes, you willing to come back together for a debrief on this lab experience? Okay. They're just cleaning up, so probably 12:35, we'll come back together.
I feel like one of those random shoot things, where it's like this. Oh, dude, history books are the worst of them. Oh, one of them. They're like, I was just confused on two of them. What? One of them was like closer. Like, they had just one what it did. Exactly. What would you I was online. I forgot why. Oh, yeah.
No.
I didn't.
There's so much, yeah.
Why are we?
You're gonna study a lot.
I'm just like putting it off.
This morning, right after.
He's adding stuff to it.
No, she,
Like,
you were
get out of the
inside,
morning.
you're still doing hard, you're still hustling really, really, really hard. But he's thinking about making it a little bit.
It's gonna be hard. And then you put home, I'm gonna be.
What?
Look at this.
Yeah.
I'm just gonna read
That's so
it.
unfair.
It's the worst. You just look it up.
And I was like,
The
I
last
just
time I
skip
got
Monday.
I got like this. I did that.
I was already, it's 2 days.
I was like, because she said something about how to use that.
Right. I just asked the folks at home this, but I'll make sure I ask you folks who here in the classroom. Would a debrief of what you just did be helpful? Just kind of summarizing the key takeaways.
Yes, please.
You're distracted, I can tell. There is a really brief lab write-up associated with this experiment. Oops, let's do 2. But I'll be very forthright. If you can pay attention for the next 10 minutes, I'm going to take you through the entire thing. It's not a claim evidence reasoning kind of write-up because this experiment is so simple. At least I hope it was. Well, the experiment itself is simple. Interpreting it can be challenging. So as I do this, if you have questions, don't hesitate to ask. But hopefully, I have this set up so that people here in the classroom can see the smartboard. people at home can see the whiteboard on Zoom, and we can all be looking at the same thing together. Okay. So under lab number 8, that post on Classroom, there's a Google Doc that will serve as your lab write-up. And again, in the spirit of simplicity, the first couple of questions ask you to consider the negative and positive controls in this experiment. So it's bringing us back quite a few weeks now, but revisiting the idea of a negative control. So remember that a negative control is a part of your experiment where you do not expect there to be a change to the dependent variable. So which tube do you think serves as the negative control in this experiment?
Tube number 1. I mixed up which one's positive and negative.
The negative control is the part of your experiment where you don't expect a change in the dependent variable. So in this case, it would be where the color didn't change.
Tube number 1.
Tube number 1? Okay, I agree. That's your negative control. Okay. Your positive control in the simple experiment, the part of your experiment where you expect there to be a change to the dependent variable. Which tube would that be?
Number 2.
Do you agree? I'm hearing 2 here in the classroom. Folks, you can pop something in the chat, or we can hear you on the speaker if you want to say something. We always welcome everyone's contributions, no matter where you are. But indeed, tube number 2 would be the positive control in this experiment, which really means tube number 3 is the experimental, right? We want to figure out whether or not we can actually hydrolyze sucrose in the presence of a dilute hydrochloric acid solution, right? All right, well then you're asked to consider, or really to draw, energy diagrams for what's happening in tube number 1 and in tube number 3. Okay. So again, spirit of review here. We're exploring the role of hydrolysis, right? We're trying to break down sucrose into its components. We can even draw those components, albeit schematically. Glucose and fructose. That's a disaccharide called sucrose. We're trying to hydrolyze it in the presence of water to produce the 2 monosaccharides, glucose and fructose. Okay? So is hydrolysis catabolic or anabolic? Are we breaking down molecules, or are we putting molecules together in hydrolysis? Breaking them down, taking them apart. So hydrolysis is catabolic, and for the purposes of these energy diagrams, are catabolic processes exergonic or endergonic? Dana is spot on. I'm not sure if you can hear her at home. Whoops. They are exergonic. Okay. So for tube 1 and tube 3, we're going to draw an exergonic energy diagram. Okay. All right. So energy on my y-axis, reaction progress or time on the x-axis. We don't know the magnitude of the energy associated with the reactants, but we know what the reactants are: sucrose and water. Like any reaction, we have to invest some amount of energy. Maybe you heard Toby asking about this earlier. To get that reaction to start. And then this is an exergonic process. The products, glucose and fructose, have less energy than the reactants do. So we've done this in the past, although it's been a while. We can label the activation energy. And excuse my penmanship. We can label the energy that is released in this process. Okay? So this is what you would draw for reaction 1, for tube number 1. And you might even ask yourself, well, wait a minute, but there wasn't a reaction. Well, yeah, there wasn't a reaction because the activation energy barrier was not surpassed. Which might surprise you, because whether you did the virtual version of this experiment or the actual version of this experiment, we heated those samples to near the boiling point. There's a lot of thermal energy being made available to those molecules in order for them to react, and yet they still don't react. That tells us something about the steepness or the magnitude of this energy of activation barrier. Okay? That doesn't change the fact that this particular reaction has an energy diagram. It's just that we weren't able to surpass that activation energy barrier to get the reaction to take place. And if it was taking place, it was taking place so slowly that there really weren't any products being formed in the first place. Let me just pause there. What questions is this bringing up for you? Folks at home, folks in the classroom. Bring you back up on camera here. You okay at home? All right. Like I said, I'm taking you through the entire lab right now. All right, so what's happening in tube number 3? Because if in tube number 1 there was no reaction, was there a reaction in tube number 3? Yeah, there was. So tube number 1 stayed blue, right, because of the Benedicts. Tube number 2 changed color, yellowish green, almost orange.
Yeah.
Orange for you folks. So orange in tube number 2, but that makes sense because the glucose that was in the solution was reacting with the Benedict solution. Tube number 3 also orangish. Okay, so there's glucose present in tube number 3. The sucrose was hydrolyzed, right, into glucose and fructose. We were able to actually visualize the products of that reaction. But why did the reaction occur in tube number 3, but not so much in tube number 1? Well, it all comes back to the presence of HCl. And let's be honest with each other, right? It's not the presence of HCl, it's the presence of H plus and Cl minus. Right? Ionic compounds dissociate freely in water, especially strong acids like HCl. They dissociate completely. All right. So it turns out that what we just observed in this really simple experiment is the role of a catalyst in this biological reaction, where H plus acted as the catalyst. And you might recall from chemistry, we're trying to tap into some chemistry background knowledge, but if I'm way outside the lines here, let me know. If none of this sounds familiar, I need to know. Catalysts lower the activation energy barrier for chemical reactions. They don't provide the energy, they simply put the molecules into a configuration which makes it more likely for them to react. So we can redraw the energy diagram for what happened in tube number 3. It would look something, again, excuse my penmanship here, like this. Where there's an energy of activation barrier, like there is for any reaction, but this barrier has changed due to the presence of the catalyst being hydrogen ion. Okay. That's it. That's the big takeaway from this really simple experiment. There are substances, in this case H+, which can act as catalysts, which will lower the activation energy barrier for reactions and get reactions to happen more quickly that otherwise would either not happen or would happen so slowly that they are virtually not happening. Okay. And for the love of dog, you do not have to know how the H+ ion mechanistically is lowering the energy of activation. Something needs to be saved for graduate school. Sometimes I wonder, in high school, are you actually taking college classes? And I don't mean like first-year college classes, I mean like third and fourth year college classes, because the people who are in college who come back to high school and visit say that high school was harder than college. What? We've got it backwards, folks, if that's what's happening. Full stop. Full stop. All right. So again, I'm talking a lot. What questions is this bringing up for you? Clear as mud, clear as ice. And degassed ice. Don't get me started with that. Opaque ice that has gas trapped in it. Okay. All right, well, that's it for this lab. We don't have a ton of time left, but the second item on the agenda was to just give you a little bit of time practicing with what you learned in the Ed Puzzle video on fat structure, on fatty acid structure. So I can actually bring you back to this, I hope. And, oops. Still in student mode, I hope. You can go to this in Google Classroom too, but just taking these last 5 minutes and practicing a bit with what you learned. I know you don't have a lot of time, but there's 2 different online interactive activities, saturated fatty acids and lipid packing, that you can use to assess your understanding of what you learned in the video. Okay. So maybe in these last 5 minutes, just choose one of those to complete. We won't get to the rest of this today. We can do some of that next time we get together on Tuesday. Finding it okay? You're quick, so I can get you over. Whatever is the best use of your time. Always come back and look at it again, get a great review. Getting up so well, appreciate it. We're gonna retake that right there. That's good. All right, folks, thanks for all your work today.
Good afternoon. Today is the last day. Have your picture taken for the yearbook. If you have not done so, please go to the auditorium and have that done today before you leave between your study period and free period. And teachers, you as well. Thank you.